Assalamu alaikum everyone. This is Yasmin Qureshi and today I will be discussing with you a new chapter from our syllabus, that is antibiotics. Antibiotics are the groups of the medicine that are used to treat the infection caused by some germs, bacteria and certain parasites. They do not work against the infection that are caused by viruses, for example common cold or flu. So the term basically antibiotics is used for the any medicine which is used against the bacteria or certain parasite. The medication which are used against the viruses, they are not called antibiotics, they are called as the antiviral. Another definition we have with us that antibiotics are the medicine that help stop infection caused by the bacteria. They do this by killing the bacteria or by keeping them from coping themselves or reproduce. The word antibiotics means against life. Any drug that kills germ in your body is technically an antibiotic. So I think you have been familiar with the word antibiotic used for the which are produced inside the bodies, okay, or the which are killing the germs, okay. Similarly, the term is used as against life, antibiotics, okay. Another thing in this definition is the mechanism of action is explained. Either it is stop the bacteria, how it is stopping the bacteria, it is killing the bacteria, or it is enabling the bacteria to cope themselves or to reproduce. So it is inhibiting the bacteria to regrow or reproduce, or inhibiting the bacteria to survive in the human body, okay. So that is called the antibiotics. So in the antibiotics chapter we have several antibiotics, and today we will be discussing penicillin. Now, this penicillin, I have referred it from the book Wilson and Griswold, Textbook of Organic and the Medicinal Pharmaceutical Chemistry, 12th edition. The link I have shared with you already. It is present on the 271 page. In this book, it says a little bit introduction for the penicillin, that the commercial production of the penicillin has increased markedly since its introduction. As production increased, the cost dropped correspondingly. When penicillin was first available, 100,000 units sold for $20, but currently the same quantity costs less than a penny. Fluctuation in the production of the penicillin through the years have reflected changes in the relative popularity of the broad-spectrum antibiotics and penicillin, the development of the penicillin-resistant strains of the several pathogens, the more recent introduction of the semi-synthetic penicillins, the use of the penicillins in the animal feeds and for the veterinary purposes, and the increase in the marketing problem in highly competitive sale areas. So these are the few reasons due to which there is the fluctuation in the production of the penicillin. These reasons are, again I'm repeating, that because of the penicillin resistant strains development, as well as the relative popularity of the broad spectrum antibiotic, as well as the production of the semi-synthetic penicillin. These penicillins are used in the animal feeds, obviously to inhibit the growth of bacteria so that animal don't die and they get the higher production of the animal, as well as it is used in the veterinary purposes. For this reason there is a fluctuation in the production of the penicillin. Over 30 penicillins have been isolated from the fermentation mixture, keeping in mind that penicillin have been isolated from one of the fungus, Penicillium notatum. So there are various penicillin which are isolated from the fermentation mixture. Some of these occur naturally, others have been biosynthesized by altering the culture medium. Commercial production of the biosynthetic penicillin today depend chiefly on various strains of the Penicillium notatum and Penicillium chrysogenum. In recent years, many more penicillin have been prepared semi-synthetically, and undoubtedly many more will be added to this list in attempts to find the superior products. This is the structure, general structure of the penicillin. You can see in this structure there are 1, 2, 3, 4 parts. First part is the thiazolidine ring. I think we have discussed this ring already before. Thiazolidine ring, because the presence of the sulfur in this ring. It is a 5-membered ring with the sulfur and the nitrogen and 3 carbon. There is no any unsaturation in this ring. Then the second part is the carboxylic acid side chain. Okay, and as well as this important part of the penicillin is the beta lactam ring. This is one of the reason that the penicillin are categorized into the beta lactam antibiotics. Okay, in the beta lactam ring you can see it is a 4-membered ring having nitrogen and the carbon. Why it is called as the beta lactam? Because the nitrogen is attached at the beta carbon. Okay, that's why it is called as the beta lactam ring. And on the 6th position we have the acyl amino side chain. Okay, so these are the important important component of the penicillin. This thiazolidine ring, beta lactam ring, this carboxylic acid side chain, and this acyl amino side chain. You have observed that only this side chain having the R in over here. It means that the various penicillin which are available, there is substitution on this position. So whenever it is written acyl amino substitution, it means there is substitution over here in the place of the R. Now let's have a look on the nomenclature of the penicillin. The nomenclature of penicillin is somewhat complex and very cumbersome. This was the reason that I haven't discussed the numbering in the previous slide, that there are basically two systems for numbering for the fused biocyclic heterocyclic system exist. First one is by the chemical abstract system. That's this chemical abstract system says that the sulfur have the first position and the nitrogen having the fourth position. Let's have the structure over here. In this structure you see opposite. Okay, why? Because the second numbering system is by the USP, and this is followed by the Wilson book, in which the number 1 is given to the nitrogen atom. Okay, and the sulfur is given fourth position. But in the chemical abstract system, first is given to the sulfur 1, 2, 3 and 4 to the nitrogen. Okay, but over here in our book the numbering is as given in this structure, so all the naming will be according to the USP. Now there are 3 simplified forms of the penicillin nomenclature have been adopted for the general use. It means you will find the different naming according to the different form. First one is that uses the term panum. Panum for the unsubstituted bicyclic system. Let's have the picture structure over here. You can see the panum is a unsubstituted bicyclic system including the amide carbonyl group with one of the foregoing numbering system as just described. Thus, penicillin generally are designated according to the chemical abstract system as 5-acyl amino over here, 5-acyl amino 2,2-dimethyl panum 3 carboxylic acid. Okay, second system more frequently in the medical literature uses the name penicillinic acid to describe the ring system with the substituent that are generally present. So you can see over here in the penicillinic acid we have the dimethyl already in the structure, as well as the carboxylic acid, and it is called as the 2,2-dimethyl 3 carboxyl. Okay, a third form followed in this chapter or in this book which we are referring, the Wilson and Giswald book, uses trivial nomenclature to name the entire 6-carbonyl amino penicillinic acid portion of the molecule penicillin, and then distinguish compound on the basis of the R group of the acyl portion of the molecule. Now you can see that this whole is this whole is taken as the molecule of the penicillin already, penicillin, and this USP1, and only this R substitution is given the numbers. Okay, so in this chapter the entire 6-carbonyl amino penicillinic acid portion of the molecule penicillin, and then distinguish the compound on the basis of the R group on the acyl portion. Okay, you can see over here also in this the numbering is according to the chemical abstract system. Sulfur is given first position, but over here according to the USP which is followed in this book, the sulfur is given fourth position. So in this book this whole structure will be called as the penicillin, only the R substitution will be named. Now this is the quick table to familiar you with the different type of the penicillins and their structure. The R group, if it is penicillin G, on the R group we have the methyl benzene structure, penicillin V, methicillin, amoxicillin, cyclosilin, and the carbonicillin. So you have to practice these structure. I know you will ask this question from me that we have to practice or not. You have to practice these structure like penicillin G, penicillin V, amoxicillin. These are very important one, as well as in the next slide we have the oxacillin and the cloxacillin, piperacillin, ampicillin. These are very commonly used, and you must have the idea how their structure is. Once you practice this general structure, the rest of the substitution will not be that difficult. Okay, so you have to practice all these structures which are given over here. I have omitted few of the structures already. If it is, I think the picture is quite clear. If it is not clear, you can refer the book. I have omitted few of the structures already. Now let's have a look on the SAR of the penicillin. First point is when the sulfur atom of the thiazolidine ring is oxidized, we have this thiazolidine ring over here. If the sulfur is oxidized to the sulfone or sulfoxide, means if the oxygen is added, it will increase the acid stability. Okay, but it will decrease the activity of the agent, means with relative to the acid it will be more stable if the sulfur is oxidized, but the activity of the penicillin is decreased. Second point is any substitution on the third position will lower the activity. So the methyl over here are very important for the activity, but any other substitution other than the methyl will result in lowering the activity. Carboxylic acid of the thiazolidine ring, if changed to the alcohol or ether, this carboxylic acid if changed to alcohol or ether will result in loss of activity. Nitrogen is important for the activity. General properties of the penicillin. The early commercial penicillin was a yellow to brown amorphous powder and was so unstable that the refrigeration was required to maintain a reasonable level of the activity for a short time. Improved purification procedure provided the white crystalline material in use today. Crystalline penicillin must be protected from the moisture, but when kept dry, the salts will remain stable for years without refrigeration. Many penicillin have an unpleasant taste which must be overcome in the formation of the pediatric dosage forms. All the natural penicillins are strongly dextrorotatory. The solubility and the other physicochemical properties of the penicillin are affected by the nature of the acyl side chain attached to, and by the cautions used to make salt and acids. Mechanism of action of the penicillin: it kills the susceptible bacteria by specifically inhibiting the transpeptidase that catalyze the final step of the cell wall biosynthesis, the cross-linking of the proteoglycan. You can also say that the penicillin inhibit the cell wall biosynthesis. How it is inhibiting the cell wall biosynthesis? By inhibiting this enzyme called as the transpeptidase. Now, the most important point regarding the penicillin, that is the bacterial resistance. Why the penicillin is no longer used? Because of the resistance created by the bacteria. There are various resistance. Some are discussed, various resistance we will be discussing very shortly, which were given in your book. First one is some bacteria, in particular most species of the gram-negative bacilli are naturally resistant to the action of the penicillins. Okay, naturally, the new bacteria which are present in present day are already resistant to the penicillin. Other normally sensitive species can develop penicillin resistance either through natural selection of the resistant individuals or through mutation. Okay, so either they are developing the other method of the resistance or through the mutation changing the forms. The best understood and probably the most important biochemical mechanism of the penicillin resistance is the bacterial elaboration of the enzyme that inactivates the penicillin. So, there are certain enzymes which are responsible for the inactivation of the penicillin. Means bacteria is producing these enzymes to inactivate the penicillins. These are by far the more important of these enzymes is the beta lactamases. The name is given just because it is inhibiting the beta lactam antibiotic or beta lactam ring. This beta lactamase enzyme that catalyzes the hydrolytic opening of the beta lactam ring. Okay, once this enzyme is causing the opening of the beta lactam ring, it results in the production of the inactive penicilloic acid. So what bacteria is doing when the penicillin come to the in contact with the bacteria, because it is inhibiting the cell wall synthesis, bacteria is producing the enzyme beta lactamases, which is causing the opening of the beta lactam ring. Once the beta lactam ring is opened, it results in the loss of activity, and that open beta lactam ring structure is called as the penicilloic acid. Okay. The well-known resistance among the strains of the Staphylococcus aureus is apparently entirely because of the production of the inducible beta-lactamase. Inducible means to cause, means the resistance is just because the production of the enzyme which is inhibiting the penicillin. Okay, the another mechanism for the bacterial resistance is the bacteria decreases the permeability to the penicillins. Okay, once the permeability is decreased, means bacteria is locking itself so that the penicillin cannot enter into the bacteria. Once it cannot enter, it cannot inhibit the cell wall synthesis, or it cannot inhibit the enzyme which is responsible for the synthesis of the cell wall. The cell that coating or that protection is the cell envelope. In most gram-negative bacteria is more complex than the gram-positive bacteria. It contains an outer membrane linked by lipoprotein bridges to the proteoglycan cell wall, not present in the gram-positive bacteria, which creates a physical barrier to penetration of the antibiotics, especially those that are hydrophobic. Now the pharmacokinetics of the penicillin. I know this chapter is huge, that's why I only discussed the important points, that is the protein binding of the penicillin. The nature of the acyl amino side chain also determine the extent to which the penicillins are plasma protein bound. Penicillin with the polar or ionized substituents in the side chain exhibit low to intermediate fractions of the protein binding, means the more the polarity of the side chain or the ionized form, the protein binding is low to intermediate. Accordingly, ampicillin, amoxicillin, cyclacillin experience 25 to 30% protein binding. The carbonicillin and the tecarcillin shows 45 to 55% protein binding because their site attachment is more polar or the ionized. Those with the non-polar lipophilic substituent Nifcillin and the isoxazoyl penicillin are more than 90% protein bound. The penicillin with the less complex acyl groups, benzyl penicillin, phenoxymethylpenicillin, and the methicillin fall in the range of the 35 to 80%. Okay, so the attachment on the acyl group is important. If that attachment is polar in the side chain, they exhibit low to intermediate fraction of the protein binding. So if the non-polar are there, the protein binding will be more. That's why the protein binding of the ampicillin amoxicillin is less, whereas the protein binding of the other penicillin like the carbonicillin is more. If we see if there are the if with the less complex, they come in the range to 35 to 80. There is a huge variation in the protein binding. But if we see if the non-polar are attached, the protein binding is 90%. Adverse effect of the penicillin include from the oral penicillin nausea, vomiting, epigastric distress, diarrhea, black hairy tongue. Hypersensitivity reaction reported are skin eruption, urticaria, laryngeal edema, and the anaphylaxis. Therapeutic application of the penicillin include pneumonia, streptococcal infections, staphylococcal infection, bacterial endocarditis, syphilis, anthrax, diphtheria, and the Vincent infection. Syphilis is the bacterial infection usually spread by the sexual contact. Anthrax is again the bacterial infection which is usually transmitted from the animals. Anthrax causing skin, lung, or the bowel disease, and that can be deadly. Diphtheria can also cause difficulty in breathing, heart failure, paralysis, or even death. Diphtheria is usually affect the mucous membrane of your nose and throat. And the Vincent's infection, that is fusous pyrocytosis, it's a progressive painful disease of the mouth that is marked especially by the dirty gray ulceration of the mucous membrane, bleeding of the gums, and a foul odor of the breath, and that is associated with the presence of the large number of the rod-shaped bacterium that is called that bacterium name is the Fusobacterium fusiform.